So this is the problem notice. If you have two reagents, that can be two type. We are try to generate a single target, or we are trying to get a multiple target. First problem I have shown that we equal to 25% HCl. That is a single target problem. If I want to make the drug discovery for a particular antibiotic, if I give you any concentration of antibiotic to you and you take it may be side effect. So actual concentration of antibiotic equal to kill the particular bacteria. So which particular concentration this bacteria will kill, that also be required the certain concentrations, a series of concentrations. Okay, so that is a multi-target. So if you have a suppose a concentration of 10% concentration of your antibiotic, and you put the bacteria, whether the bacteria is killable or not, then you 15%, then you 20%, then you 25%. This is a linear concentration. Right? And when you make it sinusoidal, when you make it harmonic, that is the different multi-reagents, single reagents, but two reagents, but multi-target is required. And this is the another one. The multi-reagents that can be reacting. That can be reacting. After reaction, you are getting some result. And that is generally single target. So these are different sampling or sample preparations possibilities. There are few works that have been already considered. Mainly based on, I must say that mainly based on IIT Kharagpur, ISI Kolkata, Professor Bhargav Bhattacharya doing this research over there. And now recently his student is an IIT, what is it? Near Uttarakhand. Yes,
Mandi.
No, Uttarakhand.
Roorkee.
Roorkee, and Taiwan, and University of Duke. This very little bit of size of research group that are working. I taking outside of it. I've just learned. So these are the basic research that already taken care. Already taken care. This is the bead scanning method, the first method that have been discovered, that we have taken 25%. So if it is the 25% or if it is the 59%, then what we do? Say the same problem HCl will equal 59% concentration. 59% concentration means 0.59. You just convert into 0.59 into its binary. By strength of 1024. 1024 is 2 to the power 10. That means number of steps you are considering 10. And what we are doing, the corresponding binary result we are taking. And first we start from right hand to left hand until the dot comes. So then when we have a sample of HCl and we have a buffer of water, so we mix it and it becomes 50%. Again we have a sample, so 1 plus 1 by 2, that is 3 by 4 something else. And then again we are doing, then again we are doing, since because this is all on. And once it is a 0, then buffer is coming.
Okay.
So you found the control word and you just to put the control word into your chip for this propagation of mixing, and finally you will get
then some sort of pulse must be there.
pulse is this is actually control word you are sending.
Okay.
And the program is basically sending the control word. Definitely a microcontroller will be there. And control word will generate this kind of concentration. Okay. And finally target is generated. Okay. So what we have actually done, we have done this multi-reagent or multi-gradient dilution for drug discovery. And recently this was actually accepted in one conference in Econ. My student is going to present this because I am a newcomer here. And this paper is already communicated to Elsevier journal in a microprocessor and laboratory. And this is one part I have given that is a harmonic multigradient sample preparation. Though we have worked with three different ways. One is a linear, as I told you, the linear, harmonic, sinusoidal, and others, all possibles ordering that are possible in a multi-target regions that we have worked. But this is a single one that we are showing here. That is basically one equation for getting the harmonic series of a given sample. If you have one sample with a 100% concentration, so if you want to get the harmonic series of n numbers, n number of concentrations, so that will be the expression. How we can do this? And whether this already solved by someone? No. Harmonic is not solved by someone, linear is solved by someone. And with compared to the linear, we calculate that much of different parameters and we compare as a benchmark. Okay. So these are the samples, concentration that we record 32 by 265. And let's see our architecture, what actually it will do the things. This will mix two droplets produced. Say one droplet is a target droplet and move to the target. And next one is not a target, but it should be wasted, but we store it. This basically storage we have used. Never this thought is considered. So we have used the storage and we actually recycled these droplets. So don't let them throw out. If it possible to reuse further, we actually take this point. So recycling we have introduced, and once we have recycled it, then we got that use advantage in this result, and then we actually reuse this. Okay. So from the recycle, it's moving over there and generating maybe target or may not be target. Okay.
Master, there is one question. Is there any difference in terms of concentration?
Yeah.
Because we are mixing.
So no, one thing that you must notice that the two droplets are stored with by a discrete one cell. So when the storage should be connected by a one unused cell, such that they cannot mix together. So the storage is also very important use, because this is the water microfluidic droplet. If two come together, by any means they can mix together. So that's why we require one barrier to hold that they can't mix together. And finally, suppose we have getting all targets, then we can always collect. And that are the results that we have get that for the different series. And from the different series that recently published by D. Mathur, that is NIT Dugamo and Professor Bhattacharya and Kaushik Chakravarti. Kaushik Chakravarti is the University of New York. Sorry, Krishnan Chakravarti, Krish. Okay. He called him as a Krish. And we compared with that publication because it's the latest one. And what we have found in a sample 74.35% we have increased because of due to use of your backup storage for
Recycling.
And buffer, 33% buffer is no longer useful still.
Now, but sir, there is a overhead of buffering is also. No? We need to take in care of that.
No, overhead in buffer is actually buffer means what? Water is freely available. So if you see in a this is basically your sample chamber. This is your waste chamber and this buffer chamber and this is the target chamber. So you can supply buffer as much as possible.
No, that can. But to store that means that brown discrete cells that you have shown. Storage cubes.
That is storage issue is the one big issue as you considered. But if you want to get the best response in terms of number of steps, in terms of waste, then you have to spend few price for storage.
Wood dhale mishti hobe.
Because the storage is now in world everything is storage dependent. So that's why not to use the storage and recycling concept. That's all. Any query? Especially to my students because those who
have you designed this thing or?
Yeah, it's a very interesting question that in India, how much progress in design? To some extent, frankly speaking, only IIT Kharagpur have one lab setup. It required huge funding. They try to produce all mixings, all those things. And especially Duke University, who is basically the inventor of this subject, Krish, Professor Krish or Krishnendu Chakravarti, he is basically the inventor of this subject. He has his own lab setup and he have filed number of patents regarding these issues. As I showed the one videos that is a prototype, that is also one patent. But again, what happened after getting the patent in 2010, 2013, another paper comes that showing that this patent is not valid. They produced some alternatives. So this is going on. But in terms of lab setups, India need to be more progressive. Since India is progressive, rather developing progressive, we are progressing. So one day Digital India concept will be successful, I hope, because this is also a part of Digital India concept. It can be useful for healthcare. And finally, therefore, our HOD sir asked me to give you the vote of thanks. I really thankful, we really thankful from Department of Computer Science and Engineering in IDAP. I want to express my thankfulness to Professor B.B. Bhuiya to be present here and demonstrate present very good. I should not say the good, very interesting cryptanalysis topic that I actually remember in my BTech or Intex classes. Really, will be helpful for our students and to our fellow colleagues, those that are undergoing the PhD works. Sambhu sir, Manish. I have queries with the Manish. Anyway, I believe Manish will not